Buenos días a todos y muchísimas gracias por compartir este día, jornada inaugural del Achucarro con nosotros. La jornada tiene una naturaleza básicamente científica, con un tono sencillo, con objeto de alguna manera, que la audiencia, que es multidisciplinar, pueda de alguna manera sacar provecho de los conocimientos que se comenten aquí hoy. Previo a la jornada científica, tenemos con nosotros a representantes institucionales que además forman parte del patronato del centro Achucarro. Como luego verán en mi presentación, el patronato es el órgano que gobierna el centro, y hoy tenemos con nosotros dos representantes del patronato. Agradecer a los dos representantes que están con nosotros la generosidad que tuvieron desde el principio a la hora de valorar la propuesta de creación de este centro en neurociencias, y en particular a Pedro Arias, viceconsejero de Educación y Universidades, que es el presidente del patronato, y a Eva Ferreira, que también es miembro del patronato en representación de la Universidad del País Vasco, de la que es secretaria general. Tiene la palabra Pedro Arias.
Decía buenos días, good morning. First of all, a few words in English for those coming from other countries. Be welcome to Bilbao, and thanks for coming to this celebration, because for us it's a very important day, the public presentation of this new research center dedicated to neurosciences. So thanks, and please enjoy this visit, not only because of the scientific discussions, but also enjoy your visit to Bilbao area. I think that the organizers can offer you not only science, but also other possible times enjoying this city and, as I would say, tourists. Bueno, bienvenidos también al resto de quienes hoy participan en esta jornada. Tal y como el profesor Matute acaba de indicar, se trata de una jornada científica dirigida a un público multidisciplinar, con la que se realiza una presentación pública de un nuevo VERC, que son las siglas de Basque Excellent Research Center, es decir, un nuevo centro de investigación creado por una iniciativa compartida entre el Gobierno vasco, más en concreto su Departamento de Educación, Universidades e Investigación, y la Universidad del País Vasco, Euskal Herriko Unibertsitatea, la universidad pública del País Vasco. No solo la universidad pública, sino también, como los parámetros bibliométricos establecen, el agente de la Red Vasca de Ciencia, Tecnología e Innovación, que produce aproximadamente el 70% de la ciencia que se genera en el País Vasco, utilizando parámetros de medida homologados internacionalmente. Para el Gobierno vasco, a quien represento en este acto, tiene una importancia decisiva la creación de esta red de centros de investigación, teniendo en cuenta no solo la captación externa de talento, como en unas ocasiones así se ha utilizado, sino también como una forma de potenciar a aquellos grupos de investigación que primero tienen una actividad pasada y presente de una calidad y de una cantidad muy importantes, pero que también trabajan en áreas que nos parecen que tienen una importancia estratégica. La historia de este centro es un paradigma de esta aproximación, puesto que dentro del Departamento de Neurociencias de la Universidad del País Vasco pues hay grupos de investigación que a lo largo de las últimas décadas han ido consolidando un currículum científico de muy alto nivel. En segundo lugar, el Gobierno, a través de la Fundación Vasca para la Ciencia, ha ido incrementando las capacidades de investigación, puesto que ha ido atrayendo a investigadores senior con carácter permanente y junior con carácter no permanente para reforzar, como decía, las capacidades de este centro. Y hoy, de alguna manera, presentamos en público el que se ha alcanzado un nivel de desarrollo, se ha formulado una propuesta que ha sido evaluada por un panel internacional de forma positiva y se ha constituido como un centro de investigación con personalidad propia, este centro vasco para la investigación en el ámbito de las neurociencias, que además lleva un nombre, el de Nicolás Achucarro, que pone de manifiesto que ya alguien nacido en Bilbao pues tuvo como científico el carácter de precursor en estas áreas relacionadas con las neurociencias, con la neurología. Yo quisiera subrayar la importancia que tiene para la Administración vasca este centro, puesto que en él van a poder colaborar estrechamente universitarios, investigadores que se sitúan en el Servicio Vasco de Salud y fundamentalmente en los bioinstitutos que se han creado para potenciar y facilitar la investigación en áreas fundamentalmente clínicas, y, por supuesto, también con otros centros de investigación, algunos de los cuales trabajan en áreas que pueden tener algún tipo de relación. Por ejemplo, pues, existe desde hace ya casi 4 años un centro dedicado a la investigación en el área de la cognición y la neurolingüística, y quizá pues, hay algún ámbito de encuentro. Decir a quienes formáis parte de este instituto que el reto fundamental para el futuro es que continuéis con esa motivación y con ese trabajo que ha caracterizado vuestra actividad hasta ahora. De alguna manera, la creación de este instituto, de este centro, crea unas condiciones en las que vuestra vida como investigadores, vuestro trabajo, pueda resultar más fácil por disponer pues, de unas infraestructuras más adecuadas, no tanto ahora, aunque ya ha comenzado, ¿verdad?, el equipamiento, sino en un futuro próximo, y también pues, un soporte que siempre la ciencia necesita más eficaz. En todo caso, finalizo siendo consciente de que este centro tiene un futuro prometedor y largo en el tiempo, puesto que se crea en un área que en este momento tiene una importancia decisiva, pero que de cara al futuro pues va a tener desarrollos importantísimos. Y por consiguiente pues va a tener una historia muchísimo más larga pues de la que tuvieron algunos bilbaínos eminentes, como Nicolás Achucarro, o en el ámbito de la música Arriaga. De alguna forma en Bilbao tenemos una historia de que algunas de estas personas más eminentes pues, no pudieron desarrollar toda su actividad porque fallecieron jóvenes. Bueno, pues yo creo que la historia de este centro va a ser exactamente la contraria. Nos encontramos en los procesos iniciales de un centro que va a tener un largo recorrido y que va a aportar a la ciencia universal muchos frutos desde este ámbito más local, pero un ámbito en el que ya colaboran personas de muy diverso origen, con distintas especializaciones, en una aproximación multidisciplinar a una serie de temáticas dentro del área de las neurociencias que tienen una enorme importancia. Y sobre todo para quienes vamos cumpliendo años, importancia decisiva. Eskerrik asko. Muchas gracias.
Tiene la palabra Eva Ferreira, secretaria general de la Universidad del País Vasco, Euskal Herriko Unibertsitatea.
Thank you. Gracias, eskerrik asko. Bueno, en primer lugar, agradecer las palabras de Carlos, de agradecimiento a las instituciones que estamos en el patronato de este centro de excelencia y, bueno, y sumarme a lo que ha dicho el viceconsejero Pedro Luis Arias, en lo que significa este centro, ¿no? Yo querría apuntar, porque poco más puedo añadir en ese sentido, apuntar que aquí, bueno, en la jornada de hoy los protagonistas sois vosotros, son ustedes, y que nosotros, desde el patronato y desde las dos instituciones que en este momento estamos aquí representadas en la mesa, lo que hemos tratado de hacer es que algo que era un trabajo difícil, en el sentido de que hay que encajar muchas piezas para que estas cosas salgan adelante, pues bueno, pues al final lo que tenemos aquí es un éxito de muchas partes, de muchas gentes. Y bueno, yo recojo las gracias en nombre de la universidad y las traslado a mucha gente que ha trabajado para que esto tuviera un buen final para empezar el ver, que es un magnífico comienzo para lo que es este centro de excelencia. Y creo que esto tenemos que ser conscientes, porque en el contexto en el que estamos, que la gente pueda hacer investigación, que las instituciones se pongan a colaborar para que esta investigación se haga, que la gente sea capaz de juntarse y formar un centro, que esa agregación se vea, que esa agregación tenga financiación, que podamos hacer estas cosas, hacerlas bien y hacerlas en un contexto que vamos a decir donde hay muchas cosas que están en la cuerda floja, creo que es para felicitarnos todos y para entender que hoy es un día especial, un día feliz, un día en el que pues tenemos que disfrutar de este tipo de comienzos, y yo en esto, pues bueno, sumarme a lo que es las felicitaciones del trabajo que se hace en la investigación de este centro, y sumarme también porque en el interés particular creo que a todos nos va mucho, ¿no?, en que todo esto sea fructífero. A mí, como decía un buen investigador de esta casa, cada vez me interesa más lo que estáis haciendo, ¿no? Pero bueno, dicho esto, pues solo me queda añadir que welcome to Bilbao, enjoy Bilbao, enjoy the workshop and have a nice workshop. Thank you.
Let me change to English, since English is the official language of the session today. So I would like first to introduce the program that we have, because one of the emphases of the Achucarro is neuron to glia signaling, and in particular in disease. So we thought that bringing the topic of Alzheimer's disease was central to the idea of the center, and for that we thought of Dr. Avila to present his own work, and I will briefly introduce him afterwards. Then we will make a journey through the other types of cells of the brain, in particular glia, and then we will have Alexei Verkhratsky, who is a member of the center, talking about how glia research originated and what is roughly the point in which we are and the challenges of understanding how they behave. And then we will go to Dr. Alfonso Araque, who is a leading scientist now in the Cajal Institute, and who is linked to the creation of this center in a way I will explain in detail in my presentation later on. And he has done a very important work in elucidating how neurons and glia talk to each other, and this will be the presentation he will talk about. And later on, we'll have a member of the center, Dr. Koen Vandenbroeck, who was, by the way, the first Ikerbasque professor to join the Ikerbasque Foundation for Science. And he will summarize what are the challenges of MS research, a disease which is typically characterized because glial cells are the target of the disease, in particular oligodendrocytes. And then I will briefly outline the strategic lines and the present situation of the center before we have the plenary lecture, which is going to be given by a pioneer of glial research, Dr. Kettenmann, to whom I will introduce before his presentation. So now let me proceed with the order, and I would like to introduce Dr. Avila, Jesus. He is a prominent neuroscientist, and he has devoted many, many years, decades, to study some of the elements which contribute to the etiopathogenesis of Alzheimer's disease. In particular, what is the relevance of the abnormal phosphorylation of tau protein, which is one of the ingredients of understanding the disease nowadays. Jesus is a chemist who doctorated in chemistry in the University Complutense of Madrid. He is a professor of the Spanish Council for Science at the Centro de Biología Molecular, Severo, Ochoa, in Madrid. And he is prominent by many means because he is also the leader, the director of the ciberNED, the network for investigating neurodegenerative diseases in Spain, a network in which we have the honor to belong to, and that harbors around 60 groups all over the Spanish geography. He has received great honors, in particular, just to name one. Yes, he's the National Prize Santiago Ramón y Cajal for biology, and has produced hundreds of papers. And we are very, very honored that he shares this day with us, because he has a very important meeting in a few hours in Madrid. But nevertheless, he managed to come and stay with us and present his work. Thank you very much, Jesus, for coming. Welcome.
Okay, thank you, Carlos. It's an honor and a pleasure to be here in the opening day for this center, new center in neuroscience. I think that also Achucarro did a lot of work in Alzheimer. He didn't know that I was doing that, but I was one of the first persons that discovered the neurofibrillary tangles, and I think that that is very important. I think that I belong to two institutions, the Center of Molecular Biology. I must say that I am not now the director. I was, but I am not now the director. And the other institution that I belong is the ciberNED. In ciberNED is an institution, is a network to study neurodegeneration in the whole Spain, and it is divide the subjects for the analysis of these neurodegenerative diseases in 3 programs. One related with Alzheimer's disease and other dementias, another one related with Parkinson's disease, Huntington disease, and other movement disorders, and the third one that is related with neuromuscular disorders. The network is distributed in 58 groups, and one of the top groups is the one that is here in Bilbao, and the leader of this group is Professor Matute, that is working, among other things, also in some aspect related with Alzheimer's disease. He has, in that program, a project related with the toxicity of beta-amyloid oligomers in neurons. I think that probably he will talk more about his own work at the end of the morning, and then I will present my own work now. Perhaps one of the main characteristics, clinical characteristics of Alzheimer's disease is an impairment in memory. One problem with the patients in Alzheimer's disease is that they cannot remember almost anything. And this thing is one of the first subjects that are under study. Why is this memory impairment? Then, to start with that, I will tell you that we know where is in the brain localized the short-term episodic memory. Because there are at least two types of memory, the episodic and the no episodic. The episodic, that is the one that fails in the case of Alzheimer's patients, is more related with facts, events, and so on. And this is the main problem in the case of Alzheimer's patients. Long time ago, I mean, in the 50s of the past century, was a patient with epilepsy that went into surgery, and the surgeon removes both hippocampal regions to prevent the development of the epilepsy. The consequence of that surgery was that the patient was unable to remember anything new from that moment. Then it was very important because this was the first signal that the short-term memory was located in the hippocampal region. And the hippocampal region, no, it's missing something here. Okay. The hippocampal region is mainly composed of two parts, the hippocampus and the dentate gyrus. I will tell you many things today about the dentate gyrus. Okay, what happens in Alzheimer's disease? In Alzheimer's disease, it seems that something similar to the problem of these patients takes place. In this case, there's also a degeneration located in the hippocampal region. And the consequence of that is similar to the consequence of the things that happens with that patients. The Alzheimer's patient starts to be unable to remember anything new. And this, the main problem, and the first type of problem is a memory impairment, a short-term memory impairment. This is one of the first problems, but we know mainly what happens with Alzheimer's patients if we start at the end. We are sure that the person had the disease in the autopsy. In the autopsy, you can see, and this is the original figure of Alzheimer's, at the end, you know that there are several aberrant structures in the brain of the patients. Some of these structures, like this, are the senile plaques. And the other ones, like this, are the neurofibrillary tangles, the tangles that Nicolas Achucarro found very long time ago. Okay, these two structures, we know today that the plaques, the main component is a peptide. The name of the peptide is beta-amyloid, and we know that the neurofibrillary tangles are composed of a protein, and the name of this protein is tau protein. And this tau protein is modified, and the modification is by phosphorylation. I mean, mainly tangles are composed of phospho-tau. And the question that was in the field for many, many years is try to see if the presence of these structures may correlate with any clinical symptom, being the memory impairment one of these symptoms. I mean, if it's any connection between plaques, beta amyloid, tangles, tau proteins, and the loss of memory. This is the question. We know that there is a possible connection between beta amyloid plaques and neurofibrillary tangles, tau. And we know because there are several studies indicating that beta amyloid, mainly beta amyloid oligomers, act like antagonists of some receptors, like insulin receptor, Wnt receptor, Fritzl receptor, and the consequence downstream of this effect is an increase in the activity of a kinase, JSK3, that is one of the main tau kinases, one of the proteins that phosphorylate tau, and that this phospho-tau is the main component of the tangles. Then is a relation between plaques and tangles, but we don't know if it's any relation between those structures and lack of memory. To study that, we are isolated a transgenic mouse that is overexpressing this kinase, JSK3, and simultaneously a reporter gene to know where this activity is expressed in a mouse, and mainly in two parts of the brain. One part is the hippocampal region, and the other part is the cortex, because these are two regions that are mainly damaged in Alzheimer disease. Then, when we start to study this mouse, the first thing that we tried to know is it was a memory impairment. We tried to do a correlation between the problems with those aberrant structures and memory. And to do that, we use this behavioral test, the Morris-Water maze, in which probably you know already, but for the people that don't know, I will tell briefly that the basis of this test is to throw the mouse to a swimming pool where is a platform. The mouse cannot see the platform, is below the level of the water, cannot see. And after throwing the animal is moving around the swimming pool until the mouse localizes the platform. The next day, when we throw the animal remembers more or less where is the platform, is looking around, and finally go to the platform. And the third day is directly to the platform. He remembers where is the platform. When we test the same thing with the transgenic mouse, we found that it takes more time for the mouse to remember for several days where is the platform. He has a memory impairment. Also, we did another test, like object recognition and so on, and we found that in this case there is a memory impairment. And then we look for the causes. What is the problem in the brain that results in this memory impairment? To do that, one student in the laboratory started to do the slices of the brain, of the normal wild-type brain and the transgenic mouse. And after looking slice by slice, he found that was a problem in this region, that is the dental gyrus. The dental gyrus was degenerated in the transgenic mouse compared with the situation that appeared in the wild type. And then we look what happens then more deeply in the dental gyrus. We found that the generation is the result of that there are less neurons in that structures. The number of neurons that is in a moment could be the consequence of the number that was previously minus the number of the death neurons, plus, in this case, the number of new neurons. This is in this case, is one of the few sites in the brain that there is neurogenesis. There is appearance of new neurons in the adult brain. Then we try to look first what is the cause of the death of neurons. And we found that one of the toxic agents was phospho-tau, because we test what happened in 3 different transgenic mouse. One that is overexpressed in JSK3, but contains a normal amount of tau. Another one that was overexpressed in JSK3, but was a knockout of tau, has no tau. And a third one that was overexpressed in JSK3 and was also an overexpression of human tau. And we found that the generation was higher in the case of that was overexpressing both JSK3 and tau, and has a lot of phospho-tau. But we also were looking what happens with the appearance of new neurons. And we know that in the dental gyrus, there are neurogenesis, and there are some precursors. You will listen today a lot of things about glial cells, and these precursors are similar to astrocytes in some way. And these precursors can start to proliferate, and afterwards could differentiate into neurons, and these new neurons could be functional, and these new neurons are involved in the new memories and in the new learning. And in fact, perhaps it's a little exaggerated, there is a person in California, Fred Gage, that suggests that a new neuron is a new idea, it's a new thing to remember. I think that perhaps this is exaggerated, but the only thing that I will try to tell you is the importance of these newborn neurons. Because if they are not proliferating these new neurons, apparently will not be new memories. That is a problem that happens in Alzheimer's disease. Then the first thing that we did is when this JSK3 transgene is expressed during neurogenesis. And to do that, we were looking for the expression of the reporter gene and to see if colocalized with the markers for the different cells that appear through the formation of the new neurons. And we found that the expression starts here, when it starts to be a differentiation of the new neurons. But to look at the process step by step, the thing that we did was to infect with a virus that is only infecting proliferating cells, this type of cells, and the consequence of that infection was the expression of a green protein, and we can follow the green protein during the whole process. This is also a method that was first used by Fred Gage, and you can see that if you do the infection and you wait for different times, you can get at different times how is the formation of these newborn cells. And you see that at the beginning, you have the proliferating cells start to have some differentiation. The differentiation proceeds, and at the end you have mature and beautiful neurons. When we did the same thing, what happens? Something happens with this, but was fine. Well, it's the most beautiful. Oh boy, that is the most beautiful. We have the other. Oh, it's a problem of. I have even. Well, I don't know if it's. I have also, but will be the same if I put.
That's another question that we could take.
I sent 2 days in advance to see that everything was correct.
Mm-hmm. So it was a slide 19th. ¿Dónde la tienes?
No, más abajo. Esta aquí. Esta. Okay. Si ponemos la anterior. Ahí está, okay. Okay, we try to do the same thing with the transgenic mouse. And then we inject the virus, and after the injection of the virus, we can see that we can found the newborn neurons just in the dental gyrus. And when we try to compare these new neurons with the neurons that are formed in the wild type, we found 2 main differences. One difference is that in the wild type, we have some kind of long neck here, some kind of Y shape, whereas in the case of the transgenic mouse, we have short neck and some kind of V morphology. Then this thing appears to be very important for the function of these neurons. We have a problem, and these are not functional neurons, and the consequence of probably that they are not functional, after sometimes they dead, whereas they continue working and continue to be functional. When we did the same experiment by doing the infection with a retrovirus that is expressing a protein that is present in the synapses, in the postsynaptic densities, also is containing the green fluorescence, we found that the spines, the dendritic spines that are the place where the synapses take place, were also a decrease in the spine number in the case of the transgenic compared with the case of the wild type. Then these features that were present in the model, we try to see if there are similar things that happen in the brain of Alzheimer's disease patients. And to look at that, we took the patients, several brains in the autopsy of the human controls and the human Alzheimer's disease patients, and we use a Cajal and Golgi typical staining method, that is the silver staining method. And with this method, we found also that there is many of these B-shaped cells in the dental gyrus of the Alzheimer's disease patients, whereas in the controls, there are more the Y-shaped neurons. And also we found that, like in the case of the mouse, was also a decrease in the number of spines in the case of the Alzheimer's disease. And then, like we are biochemists, we were looking not only to morphological aspects, but also we were looking about if there was any biochemical change in the case of the transgenic mouse. And one of these possible changes will appear in the way that neurons secrete proteins, secrete things. And these proteins that are secreted could interact with the other cells that are present in the brain, glial cells. And the study was to test for the presence of different cytokines or different chemokines that are the son of this protein that are expressed by neurons, to see how affect to microglia, because there are, for example, and I will tell you, it's not here, but I will tell you the result, is a cytokine called fractalkine, or fractalkine, that is only expressed by neurons, and when it's acting in microglia, prevents the transformation of normal microglia into activated microglia. And we found that a consequence of the dysfunction of these aberrant V-shaped neurons is a decrease in the secretion of fractalkine. The consequence of that is that microglia became activated microglia and start also to secrete another cytokines, and our studies indicate that these new cytokines could kill the precursors that are needed for the formation of new neurons. Here you can see how is the transformation of microglia to activated microglia. And the consequence of that, if we have less precursors and we have less niches, is that this is the decrease of the precursors and the decrease of the niches compared with the normal mouse, the wild-type mouse. The consequence of that is that we can deplete the precursors, and if we have not precursors, we cannot have new neurons. And this is the thing that we suggest is our working hypothesis, that this could take place also in the case of the Alzheimer patients. Our working hypothesis is that some factors like beta amyloid could activate kinases like JSK3 that could result in the appearance of aberrant neurons that die. And as a consequence of that, there is microglia activation that results in the death of precursors. And if this is repeated several times, there is a depletion. And if we don't have precursors, we don't have new neurons. And if we don't have new neurons, we have a memory deficit. Then, at the end of this, we are trying this the correlation of our first question about if any correlation between plaques, beta amyloid, tau, tangles, and memory. But we think that this, as I told you, could also take place in Alzheimer's disease. And like we are using a conditional mouse that we can increase or decrease JSK3, that is the cause, in this case, of this defect. Like we are using a conditional mouse, we can see if it's, when we go down with the cause, with the increase of JSK3, we can see if it's a reversion of the disease or it's impossible. We cannot revert the disease. And this is very important because we are using all of these models like a model also for the pharmaceutical development. If we can see that when it's not the cause, we can revert, probably we can look for a compound that will do in the pharmaceutical way something similar. And perhaps we have possibilities to do the reversion of the process. But if we are unable to do that reversion, the thing is that we must then focus in the prevention of the disease. But we will know that once that arrives to a certain point, we were unable to do the reversion. And then to do that, we started to do the treatment of the mouse. And the treatment was only to decrease the amount of GSK3 because it's a conditional model, and we can do that. And when we did that decrease in a young mouse, we can see that there is a memory recovery, and this was done using the object recognition test, sorry. And it was not only recovery of memory, but also was a recovery of the shape of the neurons. That was fantastic for us, I mean, to see that there was also this kind of recovery. But when we tried to do the same experiment in old mouse, then we were unable to recover the memory, and we are also unable to recover the shape. It was too late. Then the idea from that is that probably you try to do the treatment early, very early in the disease of the mouse at least, you can reverse the process, but you cannot do if you wait for a longer time. Then the idea is that you cannot do in the longer time because probably the problem that takes place is that there are not neural precursors, as I told you, and if there are no neural precursors, you cannot start doing the reversion of nothing because that is impossible. Then this is the problem that happens at the first steps, probably in the first step of the disease, a memory impairment that takes place in the hippocampal region. But Alzheimer's disease starts with a problem with memory, and afterwards develop, and there is a problem with dementia. And dementia is the main problem. The main problem is not the memory impairment. The dementia, that is some kind of disorientation of the patient, this is the main problem and requires help. And this is the real thing. And to look for that, we were looking how progress the disease from the hippocampal to the cortex. And one idea is that after the generation of the hippocampus, there is a neural death. And in this neural death, the intracellular proteins are present in the extracellular space because the neuron dies and then explodes. And the things that are inside are outside. And these new extracellular proteins could be toxic for the cells and could be different of some of these proteins. And we suggested some time ago that one of these possible toxic extracellular protein was tau by itself. And apparently this could happen. But there are now two different schools to look for this role of extracellular tau and how is the process. Is one school that says that first is neural death, the intracellular tau becomes extracellular, and then could be toxic to the cell, to the neuron, the surrounding neurons, because this extracellular tau, could interact with some receptors, neural receptors, that we found that could be muscarinic receptors. And that will result again in neural death, and the process continue. But there is also another laboratories that indicates that there is tau aggregation inside the damaged neuron, and these tau aggregates, could be exocytosis, could be expelled from the neuron in vesicles and could infect in some way to another neurons, and the procedure will continue for the spreading of tau toxicity without neuron death. Looking at the cerebrospinal fluid of the patient at different stages of the disease, it was found that both types of processes could take place, because it's possible to find naked tau and vesicle tau, and during the disease, the proportion of these two types of tau are possible to detect. Okay, then I will finish because my plane is almost departing now. And I would like to thank to the people that did really the work here in the Center of Molecular Biology. This is the people in the lab, and this is the external collaboration that we did. Thank you very much. Y fue fantástico, que nos arregló todo. Bueno.
Once more, thank you very much, Jesus, and good luck. Good morning, and I'm going to continue our morning presentations and our morning session with introduction into neuroglia, because obviously when we start to lay foundations to Achucarro Center of Neuroscience, we decided to study brain in its entity, and brain in its entity, of course, is composed from several types of cells, and very important part of the cell of cellular elements of the central nervous system is represented by neuroglia. And obviously the brain is probably one of the most complex organs we have encountered in our life, and presumably it's probably the most complex thing we know in the universe for the time being. And that is, of course, represents a huge challenge for natural scientists to understand how the brain works. And in a sense, sometimes ignorance is blessing, and I believe that it was a blessing for Charles Darwin, because he was very much aware that if it could be demonstrated that any complex organ existed which couldn't possibly have been formed by successive and slight modification, his theory of evolution would be in shambles. Unfortunately, Darwin, I think, did not know how complex is human brain and how difficult it is to understand how this brain evolved from animal kingdom and brought us to the possibility of making our thoughts and to make our emotions and to be creative animals in a sense. Obviously, whenever I say that, I remember another very important aphorism which was produced by Desiderius Erasmus back then in the 15th century, who said that all things in life are so multifaceted, contradictory, and obscure that we can never be sure about the truth. And that very much applies to our understanding of the brain, because so far we know really very, very little of how this very complex organ is organized. The question of how do we think and where the thoughts dwell is as probably old as mankind by itself. And what is amazing, the very first doctrine actually put the thoughts and put the cognitive action not into the head, but rather into the heart. And that was the very first doctrine which named as a cardiocentric doctrine, and for many, many thousands of years, starting from ancient Egyptians and Hebrews and ancient Greek, it was believed that this is heart, where actually thoughts are taking place. And what is interesting, Aristotle, for example, who was a big proponent of this cardiocentric doctrine, he believed that brain is a sort of a cold, very cold and bloodless organ which is needed to us in order to temper temptations and passions which are produced by a very hot organ which is heart. And the whole this theory actually started to change in about 4th century BC in Alexandria, in which the very first ever scientific institute was developed by kings of Ptolemaios dynasty, who made a museum, and that was a real institution with about 100 professors who were employed, and they had all the laboratories for different things, from astronomy to actually vivisection. And there were two people, Herophilus and Erasistratus, who made these first vivisections, and obviously that was at times without too much of political correctness and safety, so they did their experiments on humans, and sometimes even on live humans who were provided by the slavery market. And what those people found, they made a very important discovery that the brain is connected to the periphery with the nerves, so they discovered nerves. And following this discovery, they actually made a theory that our thoughts are sitting in the head. So they are somehow connected with the brain proper, with the brain tissue. And then they discovered inside the brain the structure which we know now as ventricles. And here the theory, which was called ventricular pneumatic doctrine, has been developed because what they believed, that ventricles contain a sort of air-like substance, which they call pneuma, and this substance is producing thoughts, and then this substance is distributed through the organism through a hollow nerves, and when it is released at the periphery, that triggers a peripheral reaction. This theory was very much elaborated several centuries later by Galen, who made a formalization of this theory and who actually made experiments which have been proving his own points. What Galen was doing, he was either compressing or opening the ventricles, and whenever he went to different ventricles, he found different peripheral changes, and if he went, for example, for the third ventricle, he was able to kill the animal. And his idea was that obviously when you open the ventricles, the pneuma, which is very, very light, escapes that particular part, and therefore all the thoughts and cognitive actions and functions of the brain do stop. The ventricular pneumatic theory was developed even further by René Descartes, and that was probably the person who for the first time made quite an elaborated theory of how central nervous system interacts with the periphery. So what he was believing that inside the nerves there are some small, very, very small molecules who he called an airy, fiery molecules, and those molecules could be released from nervous endings by the system of valves, and these valves have a congruent valves in a peripheral organ, like muscles, for example, which accept these molecules and trigger peripheral reactions, which in a sense was a sort of a prediction for neurotransmission as we know now. And the 17th century also coincided with the works of two very important anatomists, Marcello Malpighi and Thomas Willis, who made a next step and who started to believe that our thoughts and cognitions are connected to the gray matter of the brain, but rather than with ventricles. And the next very important step was made at the end of 18th century when Franz Joseph Gall and people who were working with him suggested that there is cortical localization of function, and that is what we know as phrenology. And here you can see a phrenological map. So what he believes that in the gray matter we have a special specialization and different parts of the brain are responsible for different functions, and that actually impairs upon the skull. So, and just looking for the skull, you can say how these particular centers are elaborated. So far, none of all this very elaborated scheme was confirmed, but nonetheless, modern techniques like in vivo imaging probably could open the way for a new phrenology, and we will know more about central localization of functions in a near future. At the very same time, electrophysiology started to develop, and Luigi Rolando was the first who stimulated the brain surface and observed peripheral reactions, and then a mapping of the brain, true mapping of the brain, started with Edward Hitzig and Theodor Frisch, who made the first electrical mapping of motor cortex, and those experiments were continued by David Ferreira, who made the first very, very detailed map of the brain. What is interesting, in the course of his experiments, he was actually convicted and almost spent 3 months in jail because of anti-vivisectionists who were protesting around his clinic, but nonetheless, British Medical Society mounted an absolutely fierce response and saved him from being put into the prison. All these efforts finished with Wilder Penfield, who in the 1930s made a final mapping of the brain of a motor cortex, which we know now as sensory and motor homunculus. All this understanding of the brain also coincided with an appearance of the cellular theory, which was born because probably of technical advance of making the first microscopes, and that was Robert Hooke, who looked into the cork, and he found that cork has a very regular structure, and this structure reminded him of the dormitories of the monasteries in which monks were living, and therefore the word cell appeared. At the same time, the first living cells were observed by Antonius von Leeuwenhoek, and here you can see his drawing of spermatozoids, which he observed by using his self-made microscopes. And this theory of cells was finally formalized by Theodor Schwann and Schleiden, who made what we now know as cellular theory, and very, very rapidly this theory embraced the feelings of all cellular biologists. At the same time, probably the first images of neural cells start to appear, and once more it was Antonius van Leeuwenhoek who was probably the first to cut the nerve and observe all these very small structures, which we know now as an axons. And then in 1830s, several people around Europe, including, for example, Purkinje, start to observe neurons and make first drawings of neurons. And then a pupil of Purkinje, Gabriel Valentin, made this drawing in which we can see already inside the neuron subcellular structures. And some other morphologists, most notably Otto Deiters, start to reveal a morphology of neural cells in the brain. And here you can see Deiter's drawings of neurons and what we know now as astrocytes. Unfortunately, he died very, very young from typhoid fever. But what is important here, all that happened before the stains, before the dyes were introduced into histology. So it was a rather painstaking work in which they sort of isolated the cells using microneedles and then made drawings. And all these things has changed after Camillo Golgi in 1870s developed the first real true staining technique, which is his very, very famous black staining or reaction or reazione nera. And here you can see his first images made in that particular time, in which you can see that this staining technique really opens the brain, and now we can image neurons and different glial cells and whatever cells in the brain in all their entity. Now, what is very interesting, you see that those images were colored. Now, it was not Camillo Golgi who made colors, but according to the legend, it was his wife who was coloring his images after he was coming home. That all led to an appearance of a grand series of how the brain is working. And probably the very first theory was introduced by Gerlach in about 1870, which was saying that all cells in the brain are connected into one functional reticulum. And that is what we know as a reticular theory of brain organization, and Camillo Golgi, for example, was a huge proponent of this particular theory. At the very same time, another very important person arrived to the neuroscience scene, and that was Santiago Ramón y Cajal, who at the beginning, by using Golgi staining, started to look into the different neurons. And what he found that neurons are not connected into the reticulum, but rather every cell is functionally separated, and integration happens when the cells are making contacts to each other with something which several years later Sherrington started to call synapses. So there were two major theories, reticular theory and single neuron theory, or reticulismo and neuronismo, which produced a conflict in neuroscience which lasted for about 20 years, and that was climaxed in 1906 when both Camillo Golgi and Santiago Ramón y Cajal were awarded a Nobel Prize. And that was quite an interesting time because their emotions ran very, very high, and both of them are produced lectures in Stockholm with a gap of about 3 days in between them, and these lectures were very much attended, and these lectures were probably extremely heated, because what Ramón y Cajal remembered in his memories of his life, that during this lecture, Golgi, in defending his extravagant lubrications, which might have been permitted 20 years ago when the nature of interneuronal connections was unknown, Golgi made a display of pride and self-worship so immoderate that it had a deplorable effect upon the assembly. And that was probably exactly what happened, because Golgi by himself, 20 years later, remembering this particular day, said, "It would have been better if on this occasion I had followed my ancient favorite aphorism, one never regret having been silent, only having spoken." Now let's put it in context. So we are talking about 1906, when a neuronal theory and neuronismo doctrine finally sort of won this conflict between two different grand theories. However, interestingly enough, 50 years before, another type of neural cell, that is glial cell, has been introduced by Rudolf Virchow, who at those days was in Charité, and that happened on 3rd of April of 1858, when he was delivering one of the lectures to the students of Medical School of Berlin, which is a school of Charité, during which he said that there is a connective substance in the brain, and that forms a sort of nerve kit or neuroglia in which nervous system elements are embedded. Now, we have to remember that Virchow, of course, was a big proponent of an idea of connective tissue, and for him, glia was a true connective tissue of the brain. Now, etymology of the word glia is still not very clear. At least the only thing we can say is that it is not coming from Greek word glue, because glue in Greek will be column. But there are several theories, and one of the theory comes from Slovenia, which associates glia with mud, and that is Slovenic word glina. In any case, all these etymological things have something bad in it, because let's say in modern Greek, the root of glia remains in the word gliodis, which means filthy and morally debased person. So somehow there is an association with glia which prevented probably us to study it in detail from the very beginning. So, just to put it in perspective, glia appeared about 40 years before the idea of neuron has come to the stage, because indeed the word neuron was introduced in 1891 by Wilhelm von Waldeyer in his series of several papers published in Deutsche Medizinische Wochenschaft. And here you can see one of the very first drawings of neuronal doctrine made by Sigmund Exner, who was a professor of physiology in Vienna, and you see that this is an essence of neuronal doctrine. Neurons are separated, and all the integration happens at the synaptic level. What is interesting, already in those days, an alternative theories which regarded the brain as a network made out of neurons and glia, and those neurons and glia are in continuous interaction, start to appear, and possibly the first person to make it, to formalize it, was Carl Ludwig Schleich, who was a medical doctor, who was a surgeon, and who introduced a local anesthesia into medical practice, for which actually he was banned from continuing his medical exercises, because people did not believe that local anesthesia can work. And in 1894, he published a book which is called Painless Surgery, Painless Operations, in which 2 chapters of this book are dedicated to his idea of how the brain works. And he believed that are glial cells which actually were instrumental in controlling the information flows through the brain. So what he believes that glial cells can change their volumes, they can shrink and swell, and when they swell, for example, the processes of astrocytes go inside, in between parts of the synapse, and therefore stop information transfer, and when they shrunk, they allow information transfer. What is interesting, 1 year later, Ramon y Cajal published another book in which he very much elaborated on the functions of neuroglia. And one of his elaborations was very similar to Schleich, so once more he believes that mechanically glial cells can intercept, can somehow penetrate synaptic cleft and stop information transfer, and Ramon y Cajal suggested that this is a mechanism for sleep. Another very interesting idea of Ramon y Cajal was that astrocytes are directly controlling the diameter of blood vessels by mechanically by mechanically controlling the diameter of these particular things. And at the same time, Ramon y Cajal came up with a very important warning, which still stands even up to now. And he said, what is the function of glial cells? And the answer is still not known, and the problem is even more serious because it may remain unsolved for many years to come until we find direct techniques to attack it. And that is a major challenge to all us gliologists, and probably during last years we got some progress, but still we are far from having a very, very selective techniques to attack glial cells. Now, definition of neuroglia, and that is from the beginning is already quite a hampering thing, because we don't have a clear definition of what neuroglia is. And most of definitions are sort of negative. So in principle, neuroglia is all cells in the brain which are not neurons and which are electrically non-excited. And that is, of course, an extremely broad term which doesn't have any... What? Does it tell me that I should stop, or what? How much time do I have? Okay. So, in a sense, we may define neuroglia as a very heterogeneous population of cells of neuron and non-neural origin, which have one thing in common: they are responsible for brain homeostasis. And of course, there are many different side types of the cells, and you know that in CNS we have macroglia and microglia, and there are lots of different cells in the peripheral nervous system, and I think our plenary lecture today, Helmut Kettenmann, is going to elaborate very much on that. There are several things which I would like to just briefly mention, and one of them is numbers. And we still don't know exactly how many cells are in our human brain, and how many of these cells are neurons, and how many of them are glia. And the notion which you can read in many textbooks that glial cells outnumber neurons by a factor of 10 is probably not correct. And overall, I believe that in human brains there is a similar number of glia and neurons. However, they are distributed very, very, very differently. And obviously we have to think why neuroglia appeared and how was the evolution, and that is going to be probably my final slide for today, but I would really like you to think about it. Because neuroglia appeared first when a nervous system appeared. And the very first nervous system was a so-called diffuse nervous system, which was composed of neurons more or less homogeneously distributed through the body of these very primitive organisms like hydra or comb jellies. And there is no indication that these organisms do have elaborated glia or have glia at all. Nonetheless, this diffuse nervous system has changed into centralized nervous system when neurons start to form a masses, start to form what we know ganglia, and start to form peripheral organs which are responsible for sensory function. And that happened at the level of nematodes. And in nematodes, there are some protoastrocytes which do assist a function of nervous cells and which do assist the sensory function, development of the cells. Nonetheless, at that stage, it's still possible to kill them all, to really ablate glia completely from the nervous system, and yet this nervous system will survive, and C. elegans, for example, would live without glia, although with certain problems. At the moment we have a next evolutionary stage, and we come to, for instance, arthropods or mollusks, neuroglia become absolutely obligatory for survival of the nervous system, and nervous system cannot live without glia because somehow the most important homeostatic functions were delegated to the cell starting from the very beginning. And what is interesting, at that stage already we have a huge diversification of neuroglia, and we have a very ancestral pre-myelin sheath, and we have an appearance of microglia. So already at the level of insects and a very, very primitive insects, mollusks, and annelids, we already have a very elaborated glia. And then all this elaboration disappears because we have another fundamentally important evolutionary step which happens at the very basis of vertebrates, of chordata, and that is appearance of layer organization of the brain. And that is connected with a complete change in how the brain develops embryonically, and that happens with appearance of a radial glia, which become a stem element of the brain and which become a scaffold which guides different cells in order to make different layers of the brain. And I think it's very important to remember, because those two types of brain, what happens, for instance, in Drosophila and what happens in vertebrates, are fundamentally different, and therefore very many experiments which try, for example, to reproduce Alzheimer's disease in fly are probably a waste of time. So at the end we came to vertebrates, and at that stage we have a huge diversification of neuroglia. We have an appearance of myelin sheath, which I believe Helmut is going to elaborate more, but that was also a very, very critical evolutionary step, because without the myelin we cannot connect all these billions of neurons with different axons. So, and here with what we have for today, and we have elaborated glia in vertebrates, and the function of these cells are still remaining quite enigmatic, and that's what I hope Achucarro Centre will contribute to understand.
Okay. Good morning, everybody. First of all, I want to thank Carlos and the Achucarro Centre for inviting me to participate in this event. I think it's very exciting, and we have to congratulate ourselves in the neuroscience community that this center devoted, I think, is one of the pioneer centers in the world, probably the first center in the world devoted to the neuroglia interaction. We are very happy, and we have to congratulate ourselves that this is created in this beautiful city. And I wish this center a long life, as it has been said before. And I think today is going to be, for the future, one of the landmarks of the neuroscience community in Europe and in the whole world. That's my wish about that. So when Carlos invited me to present here my work, I decided to talk about how neurons and glia talk to each other, which is the main focus of the work in my lab. We have heard a wonderful presentation by Alex about these glial cells, and I want to present very briefly, with no much details, about how these glial cells, and specifically astrocytes, are interacting with neurons to actually make our brain function. That's our working hypothesis, that our brain is not the result of the neuronal activity, but the neuroglial activity. In a coordinated way, they talk to each other and they perform the function of the brain. And I'm going to present some basic and sketchy ideas about why do we think that way. I did my PhD at the Cajal Institute in Madrid. Cajal was the mentor of Nicolás Achucarro, so it's very close to I could be then, if I did my PhD in the Cajal Institute, I have the grandson of Nicolás Achucarro in the scientific point of view. So I did my PhD in the Cajal Institute, and I want to present this image, this drawing. Okay. This drawing made by Cajal. You know, Cajal was one of the greatest neuroscientists, but he was also a very good artist. He was able to produce this kind of wonderful drawings. You know, Cajal is very famous by his studies on neurons, but he was also very interested in glial cells, and he devoted a lot of work to the study of glial cells. And you can see in this picture how it was taken from a human hippocampus, and you can see the center of this drawing is an astrocyte. This is a specific type of glial cell. The neurons are kind of faded away. He was not interested at this point in neurons, so he wanted to stress the importance of astrocytes, and he put the astrocytes in the center of the image to convey the importance that he was considering about glial cells in the brain function. There are many different things here in this drawing about our brain. You can see here the first demonstration that astrocytes, glial cells, can divide in an adult person. So it's the first demonstration here. Out of this static image, he was able to think that glial cells could develop in the adult, unlike neurons, that they do not develop, although we have seen, as Jesus Avila was showing before, that they do in the neurogenesis. But at that time, it was considered that neurons could not divide, but astrocytes were able to divide. They were also contacting blood vessels, and you see here, as the drawing that Alex showed before, how astrocytes are in intermediate positions to conduct the blood vessels and the neuronal activity. And we know now that this is a very important function of glial cells, and specifically astrocytes, how to control the brain microcirculation. But what is important for me in my studies is this kind of image, that you can see a neuron here and an astrocyte, which is big and a big hug to the neuron. They are just showing the intimate relationship between astrocytes and neurons. They are very good partners. They stay together and they hug each other. Like I said, this image is kind of idealistic image drawn by Cajal, but it's not that real. This is the reality. Here you get the concept of the center of the astrocytes and the importance of the astrocytes. But this is taken from a colleague in the Cajal Institute, showing a neuron, now from a rat, and a small astrocyte surrounding that neuron. They are much smaller. It's not as bigger as this, but this is much smaller, with small processes around that cells. But still, this intimate connection is still present there. They are very close together. And for more than 100 years, they were considered that astrocytes were not playing any role in brain function beyond the homeostatic control of the neuronal activity. And why what happened that way, that for many years was not considered astrocytes an important cells? Probably because we know from the 16th century, 17th century, that the brain is an electric organ. The brain functions with electricity. Our neurons work with electricity, and electricity is important in brain function. But as Alex said, astrocytes are electrically non-excitable. They don't have excitability. If you record electrophysiologically from a neuron, you can see action potential, you stimulate that neuron, and it responds with action potentials, with electrical pattern activity. But if you record from an astrocyte, you get totally bored. They don't do anything. They are just electrically excitable. And probably for that reason, we don't know why, but probably for that reason, they are not excitable, and our brain is electrically excitable. Then probably is that the reason why for almost 100 years we thought that our brain function results only from the activity of neurons. But this idea became challenged in the 1990s when the new methodologies for imaging intracellular calcium in living cells were introduced in the study of the neuroscience. And from two pioneer works published in the early 90s of the last century, we could observe that astrocytes, which indeed are not electrically excitable, they can be excitable based on intracellular calcium bases. Here, there is a carpet of culture cells, astrocytes, grown in culture. They are taken from the brain, and they are put in the culture, and then we could monitor the intracellular calcium levels of those cells. And there are many, many cells here. One of these is cells, and we are going to stimulate one of them, and we are monitoring the calcium. And you can see what happened. Here you are. You have an increase in the intracellular calcium of that cell that we stimulated, and this calcium, this signal, also communicates with the other cells, forming this wave of calcium in the culture. So the astrocytes are not electrically excitable, that's right, but they are excitable based on intracellular calcium variations. And even more, these astrocytes communicate with each other. They can talk to each other. We have a lot of astrocytes in our brain. Okay, then they could communicate with each other, just like neurons. So they have electrical excitability, and they communicate with each other. And they show this calcium excitability, and this calcium excitability can be, and you monitor in the brain, in slices, you can monitor, and you can see that this calcium excitability could be spontaneous. The astrocytes show that calcium variations by themselves with no external input, just by intrinsic properties. But in addition to that, and this is important, they also can be evoked by the synaptic activity. But when two neurons are communicating to each other, they can trigger this signal to the astrocytes. I'm going to show you here a movie, and you can see 3 astrocytes, and we were stimulating, in this case, the axons arising to that area, and we monitor the cellular excitability of astrocytes, and you can see that after the stimulus, there was an increase in the calcium. So astrocytes are excitable, and they can listen to the activity of the neurons. When neurons talk to each other through the synaptic activity, they are informing to the neurons, to the astrocytes, what is happening. And astrocytes can listen to the neurons. We know now that this is happening in many areas of the brain, and these calcium changes in the astrocytes are mediated by different transmitters that neurons release to communicate with other neurons. But at the same time, the same transmitters are able to communicate to the astrocytes and to talk to them. So neurons talk to astrocytes, and astrocytes listen to the neurons. And this, I show you, this was done in brain slices. But this not only happened in brain slices, they also happen in vivo. If you record using a two-photon microscope, or in our case a confocal microscope, in a living mouse, you have a mouse and you put the mouse below the microscope, and you can monitor the astrocytes there. You can see here, it's not very clear, but we could monitor the astrocyte here, and we could monitor the calcium levels of those astrocytes in the brain, in a living animal which is breathing there under the microscope. And then we stimulated, in this case we stimulated the tail. We just pinched the tail. We created an stimulus, an external stimulus, and we observed that in most of the astrocytes there was a calcium elevation in the astrocytes. The astrocytes were responding to the activity of the stimulation of the body of the astrocytes. So they are sensing, through the activity of neurons, they are sensing what is in our external world. And this is due to the ability of astrocytes to respond to the activity of the neurons. When we stimulated this tail, there were neurons there receiving information, that carried information to the brain. And when they are talking to other neurons in the brain at the same time, they were talking to the astrocytes and evoke this calcium elevation in the astrocytes. So astrocytes are able to respond to the neurons and also to the sensory stimulus. But they not only respond to the neurons, they are not like the servants to the neurons. The neurons talk to them and they obey. No, they can also talk back to the neurons, and they can modulate and regulate what neurons are talking to each other. I'm showing here is an example of the work I did years ago when I was in Iowa in the United States, in the Phil Hayden's lab, just showing that when evoking this calcium wave in the cultured astrocytes, and if we monitor the synaptic connectivity of two neurons, how two neurons are connected, showing this kind of synaptic currents, which represents the connection between two neurons. You can see that after the stimulation of the astrocytes, the amplitude of this synaptic response was transiently reduced by the astrocytes. So astrocytes could talk back to the neurons. When they increase the calcium, they could send some signals and then modulate how the neurons talk to each other. Actually, we know from the work of many different labs that this talking back to the neurons is mediated by the release of what it is called gliotransmitters, which are like the neurotransmitters that neurons use to communicate each other. Astrocytes also can release these neurotransmitters, but we call them now gliotransmitters. And these substances, these molecular signals, can produce different effects on the synaptic transmission and the neuromodulation. And we have seen that, not me, we, neuroscientists, we have seen that in many different parts of the brain are mediated by different gliotransmitters released by the astrocytes. They could release several molecules that they can produce multiple effects on the way that neurons are active and the neurons talk to each other. They can therefore control how neurons talk to each other. Actually, in an example of this phenomenon, I want to show you this kind of example that in a single synapse, the single specialized structure, very small structure in our brain that is the interface of communication between two neurons, astrocytes can modulate the efficacy of that specialized structure. You can see here, we were recording the activity of the postsynaptic cell. We were stimulating a single synapse in that preparation, and we were stimulating a single astrocyte, elevating the intracellular calcium. By methodology, I'm not going to enter, but I'm going to show you here how we could control the calcium in the astrocytes, and you can see after the stimulus we deliver to the astrocyte, we evoke this huge calcium elevation in the astrocyte. When we're recording this connection, this connectivity in that synapse, the strength of that connectivity in basal conditions, we observe that in most of the cases there was a failure in synaptic transmission. This is something that happens in our brain, that synaptic connection between neurons is very, very inefficient. If it was designed by an engineer, he would be fired, because it's very, very bad connection. You see, most of the times, neurons fail to talk to each other. That's very important for our brain, but it's a very bad design. But when we elevated calcium in the astrocyte, you can see the number of failures and is decreased with respect to the control. but the number of responses in that case is increased. That means that an astrocyte can regulate the efficacy that is connecting two neurons between them through the synapses. They can regulate that efficacy, which is very important for the information processing in the brain, how the synapses are, the efficacy of the synapses. As Jesus Avila introduced in the first talk, there is an important phenomenon in neuroscience which is called synaptic plasticity, which is the basis of the learning and the memory in our brain. Synaptic plasticity is the phenomenon that makes that the synapse, the strength of the connection of a synapse can be regulated and can be changed, altered over time, for a long period of time. And this synaptic plasticity phenomenon are recognized as the cellular basis of the learning and the memory. And therefore, it's devoted a huge amount of work by the neuroscience to understand the processes and the consequence, the pathological consequence of those processes. And I want to show you here that the astrocytes can actually induce synaptic plasticity changes in the synapses. You see here, when we stimulated a single astrocyte and we stimulated also a postsynaptic neuron simultaneously, we could evoke the long-term potentiation of that synapses. The efficacy of connection between the pre and the postsynaptic elements in that synapse could be increased for more than 60 minutes in our preparation, but in other conditions could be days and weeks. Astrocytes could change the synaptic plasticity, could change long-term the synaptic strength of that synapse. This, like I said, this is a very new or recent topic, how astrocytes could be involved and how could regulate synaptic plasticity, because it would be a new way to understand the cellular basis of learning and memory. And this has tremendous impact in our concept about pathological states when there is an impairment in learning or memory. And this modulation by astrocytes of synaptic plasticity has been demonstrated relatively recently in different areas by different endogenous stimulus, different stimuli to the astrocytes, and using different gliotransmitters to modulate that. But again, this could be just a pure artifact of in the different laboratories when we're using brain slices or culture cells, but that's not the case. This could happen in vivo. Astrocytes can induce synaptic plasticity in vivo. What we did recently was to monitor, like I showed you before, the calcium levels in the astrocytes when we were stimulating the tail of the animal. And we were monitoring the calcium levels in the astrocytes, and we could see an increase in the calcium in those astrocytes. And also, in the parallel experiments, we recorded the activity of the hippocampus, that region of the brain that Jesus Avila talked about and that is known to be involved in memory. And we record the activity of the synaptic transmission in that area when we were stimulating that, the tail of the animal. And we observed this thitherine, the special rhythm in that region that is involved in the generation of the long-term potentiation, that synaptic plasticity in that region. And this synaptic plasticity in that region could be blocked, was abolished in vivo when these calcium elevations in astrocytes were abolished. If we did not allow the calcium in the astrocytes to elevate, then there was no synaptic plasticity. There was no memory in that animal. So astrocytes can induce synaptic plasticity in vivo through a very complicated way that I'm not going to enter today. So, in summary, the evidence provided by many different laboratories throughout the world, including ourselves, is that astrocytes exhibit a form of excitability based on intracellular calcium variation. It's not electrical excitability, but it's a calcium-based excitability. That astrocytes can respond to neurotransmitter released by the synapse, by the neuronal activity. When neurons are active, they talk to the astrocytes. And in turn, the astrocytes can release gliotransmitters So they talk back to the neurons, and they regulate the activity of the neurons. They regulate the way that they connect to each other, the way that they talk to each other. So they are just like a group talking to everyone. And then astrocytes, through the release of gliotransmitters, can modulate this synaptic transmission and this synaptic plasticity. And these evidence have led to the establishment of a concept in the synaptic physiology, a new concept which is called the tripartite synapse. That the synapse are actually functionally, the function of the synapse is constituted by three elements. Not only the presynaptic and postsynaptic neuronal elements, but also that the adjacent astrocytes, that they are forming a unit, a working unit. And that's one of our proposals for the basis of the brain function, that these tripartite synapses are actually the functional units for information processing in the brain. That's what we try to demonstrate in the future. So you could say, okay, this is nice, maybe I convince you, this is nice, but this is animals, and this is just experimental preparations. Does this happen in our brain? Is in our brain working with astrocytes? When we recall something about 20 years ago, are we using astrocytes or only neurons? Well, to try to address this question, which is not easy, we're not allowed to work with humans yet, or not anymore, as Alex said before, not anymore. But we used brain slices from epileptic patients. These patients, in some cases, the only way to treat these patients to prevent the epilepsy is to remove, as we have seen before in the Jesus Avila talk, remove part of the brain. It's the only way that these patients can live more or less in a proper life. So the surgeons give us this tissue extracted from living patients, adult patients, living patients, and they give it to us. And we record from that tissue. And I can show you here, this is an hippocampus from a human being. And you see here some astrocytes and neurons, and when we stimulate here, I'm going to stimulate here, these astrocytes, and you can see, it's not very clear, but you probably can see, not yet, again, too low. You can see the calcium elevation in that region, and it goes like a wave that also involves the neurons there. Actually, if you record the electrophysiology from one of these human neurons, you can see, you can detect the gliotransmitter released by the astrocytes in the form of these slow inward currents in the neurons. So the human astrocytes display spontaneous calcium oscillations, the same thing that we have seen in other animals, experimental animals. Neurotransmitters can evoke associated calcium elevations, and these calcium elevations can induce the release of gliotransmitters that can modulate neuronal activity, activating neuronal receptors. So there is a bidirectional communication between astrocytes and neurons in the human brain tissue. It's not something specific of the experimental preparation. It really happens in our brain. And probably the way we think, the way we remember, the way we recall something in the past is using not only neurons. We're using neurons, of course, and for sure, but maybe we're also using astrocytes. And I just want to finish with this slide, just showing I was showing something about the physiology, what we believe astrocytes are doing in the physiology. But this may be very important, and I think the Achucarro Center is going to be one of the centers that devoting to the neurodegenerative diseases to show how important is this astrocyte-neuron communication. The interactions could be involved and could represent the basis for many neurological and neurodegenerative diseases. Most of the studies done so far have considered, regarding neurological and neurodegenerative diseases, many of them just focus on neurons, what happens with neurons. But if we are aware now that both astrocytes and neurons play together, dance together, work together, and the brain function results from the interaction of both types of cells, maybe the disorders in the function of the brain results from the disorder in the communication between these two types of cells. This is something that has to be addressed in the near future, and I think the Achucarro Centre is in a very well positioned to undertake that role and to show that that could be a possibility. And they could probably identify, they will serve to identify new targets to the treatment of these different diseases. So the summary, which is a kind of a bold statement, and you will indulge me for that, but it's a kind of not only what we know, but also what we would like to propose, or we would like to think, or what I like to believe, is that there is a bidirectional communication between astrocytes and neurons. That the astrocytes are actively involved in the integration, transfer, and storage of information in the brain. They are not only just passive bystanders to see what neurons talk to each other. No, they are really actively involved in that conversation between neurons. And therefore, the brain function results from the coordinated activity of neuroglia networks, not anymore neural networks. We know that about neuronal networks is provided. That was the whole idea. But no, the idea is that the brain function actually arises from the coordinated activity of neuroglia networks. And that neurological and neurodegenerative diseases may be related to dysfunction of this astrocyte-neuron communication. And this opens a new view for the future about how to treat these diseases. And they could be very important in solving these problematic disorders, brain disorders. I just want to finish with the people in the lab and the collaborations and the funding institutions. Thank you very much.
Vínculos, si quieres, dale. Para que esa parte la tengamos. No son. ¿No es ese? Ese no es. Igual tarda un poquito en abrirlo porque como estamos teniendo problemas. Pues sí, tarda así. To introduce yourself to use the pen. Well, Carlos was supposed to do. Okay, so the next speaker is Koen Vandenbroeck. He will speak on multiple sclerosis and his approach, or the approach that his group has on this neurodegenerative, I would say, disease. Sorry, Koen.
So thank you. And now for something entirely different, but multiple sclerosis, genes and biomarkers. But first of all, I would like to thank Carlos for his invitation for me to present this talk, and I would like to express also my appreciation to the Basque government, Ikerbasque, and University of the Basque Country for the creation of this center. We may be a small regional center right now, but I'm sure with a lot of hard work and dedication, we will grow into a national and international reference. what is multiple sclerosis? A chronically inflammatory disease of the central nervous system, which is characterized by demyelination and axonal degeneration. The disease can present itself in different forms. The more common form, relapsing remitting MS. Another form is primary progressive. It's also more common in women, and it shares that characteristic with many autoimmune diseases, such as type 1 diabetes. It's a prevalence of 0.1 to 1.5 per thousand, and so far there's no cure, only disease-modifying treatments that limit the progression of the disease. What is the cause of MS? Multiple sclerosis is considered a multifactorial complex disease with a modest genetic risk heritability, and displays also multifaceted interactions between genetic variants and environmental factors. The last years we have seen really a tremendous progress in the characterization of genome variants that predispose to multiple sclerosis, and also some environmental risk factors have been characterized pretty well. So my talk will focus on these two groups of factors that cause MS. There may be other events involved, epigenetic factors, but so far progress is rather limited, so I won't cover it in this talk. The story of the genetics of MS started in 1972 with the discovery of HLA as a risk factor for the disease. HLA-DRB115 haplotype that's been confirmed many times and refined over many years. And the point is that when you carry this allele, you have a highly increased risk to develop the disease. However, there are many MS patients who don't carry that allele and still get the disease. So on the basis of this and additional observations, it was clear that there were other genes involved in causing multiple sclerosis. But it took a very long time to develop the models, to develop the collections, the strategies to identify these genes. And actually only in 2007, with the arrival of genome-wide association studies, the first risk factors for MS were identified, 6 of these. Since many more genome-wide or GWAS studies have been performed, and right now, with the latest study published in August in Nature last year, the total number of risk genes for MS is 57. I won't review all these genes, but what is important, if we look, and many of these genes, well, most genes we know pretty much about their function. There are some genes in which we have no clue at all. And we look at the function of these genes that we know the function of, it appears that they all play a role in the early stages of an immune response in antigen-presenting cells, also in the early differentiation of Th0 cells and Th1 and Th17 cells, which are two branches of the cellular immune response, which are considered very important for MS and lead ultimately to tissue damage. GWAS is not the only instrument to find risk genes for MS, and GWAS is very powerful. Of course, it's model independent, allows you to screen the whole genome, but the commercial available arrays for GWAS don't cover equally all areas of the genome. So candidate gene approaches are used as well, and that's a strategy we used in our lab. We screened 55 genes for cytokines and other immune-relevant molecules. We did that in the Biscaya cohort, which is a cohort of 650 MS patients encountered here from Basurto Hospital. And we identified one factor, SOX, suppressive cytokine signaling 1, an anti-inflammatory factor that was very strongly associated in this cohort. You can see here, I don't know if it's very clear, but p-values 10 to the -7, odds ratios which are consistently higher than the average odds ratio found for non-HLA risk factors. We also validated this association because it's very important. You can find an association, but it may represent a false positive. You need to validate that. And we have validated that in many more collections, so we are pretty sure right now SOX1 is a susceptibility gene for MS. And independently, another group from Vanderbilt University found this gene using a different strategy. Another gene we found is ankyrin D55. It was originally identified in rheumatoid arthritis, and it is known that several genes are shared between autoimmune diseases, right? So on the basis of that hypothesis, we decided, together with the Spanish network of multiple sclerosis, to investigate some of these genes. And ankyrin D55 arose as a gene with high significance and an odds ratio of 1.26 in Spain. In order to make sure, to be absolutely sure that this indeed is a susceptibility gene for MS, we did an additional validation in a German and French cohort with collaboration with Frauke Zeb and Christina Lill in Germany, which confirmed the association. So pretty sure of this gene, and part of the work in our lab will now be focusing on exactly, we don't know the function of this gene, what it exactly is doing. Once you have identified a risk gene for MS, studies are needed, right? We need to understand why polymorphic variants in the gene, what are they exactly doing? How did it change maybe the function of the gene so that it becomes a factor of a risk factor for multiple sclerosis? The first stage you normally do is fine mapping, and that's what we did for CD6. CD6 was not discovered by us, but it appeared significantly associated here in the Biscaya cohort. So we did a fine mapping with 14 SNPs, and we're able to find a haplotype, a combination of markers that all change amino acids in the extracellular domain of this receptor, and which confirmed that in 3 more cohorts from Madrid, Andalusia, and UCSF. We performed a detailed flow cytometry analysis in which we looked at 15 different lymphocytic cell categories, and we found that the genotype in this gene, the combination of markers that I mentioned, strongly affects expression of CD6 in naive T cells. And also the high-risk haplotype is associated with high interferon gamma production, a very important pro-inflammatory cytokine. Together, our work seems to indicate that CD6 plays, and very confirmed the model I showed you before, plays a role in the early stages of immune activation in multiple sclerosis. So that's where we are now. Many more studies will be appearing over the next years, and it is likely that the number of total MS genes will go up significantly. The challenges, we need to do much more fine mapping. The SNPs, the polymorphic variants, which we found to be associated, are not necessarily the ultimate causative variants. There may be also rare variants that have high penetration and have drastic effects on protein function and structure. And in order to find this variant, we will need to do genome sequencing, or at least exome sequencing of the coding regions. But so another challenge is to integrate these genomic data with transcriptomics, proteomic, epigenetic, metabolomic, and environmental data in order to describe multiple sclerosis, immune pathogenesis, and MS as detailed as possible. There's also great interest in understanding more about the clinical course of the disease and why some people suffer from a more severe course of disease or benefit from a more benign course of the disease. There have been GWAS, genome-wide association screens, to find genes, but so far it's been quite unsuccessful. Regarding environmental factors, there are now 3 factors that have been established firmly as risk factors for multiple sclerosis. Yeah, this came a bit bad, but so first smoking. The study of smoking in MS started in 1965, and many studies have appeared that have been published that show indeed an increased risk for multiple sclerosis in people that smoke. We know in the meantime also there's not a nicotine that is the wrongdoer. And why we know that? Because people that use snuff tobacco have no increased risk for multiple sclerosis, and one study even has demonstrated they have a decreased risk for multiple sclerosis. So more likely it is the smoke particles that damage the lung epithelium, causing recurrent infections or autoreactive T cells that may potentiate the autoimmune response in multiple sclerosis. There's a strong dose response here. The more you smoke, the higher the risk. Smoke is not only bad to increase your risk for MS, but it can also lead to a more progressive disease, and you will acquire a more rapidly disability when you smoke. The mechanism is unknown, and maybe it's important to look at this because there's indications that young women right now are relatively smoking more than young men. And young women are also more at risk to contract the disease. So understanding how smoking interferes with genetic factors and the full mechanism will be an important challenge. Over the years, many viruses and infectious agents have been associated with MS. But firm evidence is now in place that Epstein-Barr virus infection is a contributing factor. EBV infection is found in 94% of the human adult population. But in multiple sclerosis, it reaches virtually 100%. In addition, the risk for MS is 2 to 3 times higher in individuals that have symptomatic EBV infection, meaning infectious mononucleosis. And it's 20-fold higher than EBV-negative subjects. This is very strong epidemiological evidence, of course. The mechanism of how it works is virtually unknown. I passed this graph here to give you an indication, but it's only covering small parts of the puzzle that is Epstein-Barr virus infection in MS. Let's not spend too much time here. The important question that we need to answer right now is what is the mechanistic underpinning of the epidemiological association between MS and EBV? And also, can we introduce alterations in the immunobiology of EBV to target for MS therapy? Everyone who is a little bit familiar with the geographic distribution of multiple sclerosis knows that the incidence and prevalence of the disease is particularly high in the northern hemisphere, coinciding with the Western so-called civilized world. And it's firstly, if you look at this map, the lower map here, which gives the human skin color distribution, shows that the pattern is opposite, right? You see more prevalence of MS in areas where people have lighter skin. So we know that the incidence and prevalence of MS increases with the latitude of childhood. And this latitude gradient correlates inversely with the level of ultraviolet radiation exposure. And we know that UV light is an inducer of vitamin D, and this led to the hypothesis that vitamin D, one way or another, influences risk for MS. There are additional indications that vitamin D indeed plays a role here, such as that MS patients are more likely to be vitamin D deficient prior to onset of the disease, and also that MS risk is increased for late spring births when the level of vitamin D is pretty low. Vitamin D, how exactly vitamin D works in MS is not known. We only have fragmentary evidence on its effect on various categories of immune cells. It's like a decrease on pro-inflammatory production of cytokines, MHC class 2, plasma cell differentiation and secretion of antibodies. It comes out poorly here. Okay. Okay. This slide aimed to clarify something. Anyhow, but what I wanted to show here is we talk, and I indicated in the beginning of my talk, multifaceted interaction between genetic factors and environmental factors, and this is particularly very well recently demonstrated for vitamin D because 3 genes have been discovered that are actually coding for enzymes that catalyze different steps in the synthesis pathway of vitamin D that acts as risk factors for multiple sclerosis. And one of these genes is now, for one of these genes, really very strong evidence has been obtained that the CYP27B1 gene, which contains mutations that change amino acids, leading to loss of enzyme activity, is associated with lower levels of calcitriol, the active form of vitamin D. And in one study done by George Ebers and colleagues, it was shown that these mutations were always transmitted in families to the affected patients and never to the non-affected members of the family. So therefore we can say that vitamin D is an environmental factor that is associated with protection against multiple sclerosis, but that genetic modifiers may modify the level of vitamin D and this way modulate the risk for multiple sclerosis. And this, of course, needs to be described now much, much better with epidemiological studies, both performed in northern and southern locations, to assess the relative contribution of genetic and environmental factors. So it was a very brief overview of MS risk factors, environmental and genetic, and of course, in the long run, what we hope to obtain with this kind of studies is new therapeutic windows to interfere with the disease, a better knowledge of the disease, and ultimately therapeutic opportunities. Already, right now, we can already some of that information in order to try to predict the risk for multiple sclerosis. There have been several studies performed in this regard, and the study that probably is more similar in this regard is that of Philippe de Jager, who developed an algorithm based on the weighted genetic risk score on 16 MS risk genes. Right now we have many more, right? But this was based on 16. And of course including the HLA-DRB1, and he found that the algorithm was quite robust, but had not a very strong predictive capability. To give you an indication, the C statistics of this algorithm were 0.72. The framing heart score which is used for coronary heart disease is 0.8. It's not so far, but apparently we will need more integration of more risk factors in order to make this algorithm really workable. For now, it can be used in order to predict risk to develop the disease in first-degree relatives of MS patients. So predicting genetic risk of the disease is one important question. There are many other questions that scientists are aiming to answer via the identification of biomarkers. Biomarkers are molecules or characteristics that can be quantified and evaluated and are indicators of normal biological processes, pathogenic processes, or, for instance, the pharmacological response to a certain drug treatment. And here are several questions that let's say, scientists are hoping to address via the identification of these biomarkers. And one question that is of particular importance to our lab is a pharmacogenomic biomarker question. There are several drugs being used for multiple sclerosis right now that work or are capable of delaying the disease progress, but they don't work in a considerable proportion of MS patients. The question is, why is that? If you could find biomarkers that would give us information prior to about the probability of a person to respond to the treatment, prior to the onset of the treatment, we could already avoid a lot of difficulties. We don't have the necessity to give the drug and just wait for 2 years until we can make a balanced decision on whether or not the patient is responding to the therapy. Different categories of biomarkers here, diagnostic biomarkers that allow to discriminate MS patients from healthy individuals or other patients with neurological diseases. Predictive biomarkers that could help to determine the risk of conversion to MS at the time of clinically isolated syndrome, because not all people who suffer that will convert to multiple sclerosis. It's an important question. Process-specific biomarkers are biomarkers that are related with specific pathophysiological processes, such as inflammation and demyelination, oxidative stress, glial activation, and so on. And the idea would be here that this kind of biomarkers could be related to disease activity on an organism level. And then finally, treatment response biomarkers. Here I've summarized some of the important biomarkers found already. Many of these, or actually all of them, need validation, right? When you screen the literature, you see that, that there's an increasing reports on biomarkers in MS, but there's very little overlap between studies in terms of design of the study and also in the identification of the biomarkers found. So this will be an important challenge for the future, validate on a biological and clinical level the biomarkers found. I will conclude here my fast talk and just propose my group, the neurogenomics group, all people who have been working hard in order to advance our research. Special thanks also to Dr. Alfredo Antigüedad, the head of neurology department of Basurto Hospital, who supplies us with MS biosamples and clinical data. And in order to validate genetic associations in other collections of MS patients, we can count on the generous support of various investigators both in Spain, in Germany, and in the US, which I've listed there. Thank you for your attention.
We are a little bit behind schedule, so we apologize for that. And we also planned the whole session without interruption because we wanted to invite you for lunch at the end. So, yes, thank you for your patience. So, my presentation will deal with the organization, with the Achucarro Centre. And we chose this title, Knowledge to Cure, because we want to emphasize the importance of basic and fundamental science to have biomedical applications. In other words, no cow, no milk. So this is very important because often this idea is not emphasized enough. So I will make a brief outline of what the center is and what it plans to do in the near future. So the synopsis is, firstly, I would like to explain you how the idea developed and crystallized to, until today, some basic concept about the strategy of the center, an important body of the center, which is the advisory board, and of course, which are the groups and what they do, and what kind of facilities and equipment we have. And just to make the story very brief, we soon realized after the Ikerbasque Foundation was created that bringing scientists to the Basque Country, and namely bringing neuroscientists to our environment, was perhaps not enough, that we would need an organization to add value to the initial proposals that scientists were bringing to our environment. So the Basque Foundation for Science is now 5 years and a few months old. So the first talk and discussions occurred more than 4 years ago and were initiated in a small meeting we had in our laboratory with colleagues from Europe, among them Alexei Verkhratsky, who is here today, and a member of the center. So conversations with the administration were very positive. The initiative was well regarded, and we were invited to submit a full proposal. And we did so in November 2008. And when I say we, I mean Alexei Verkhratsky and myself. Unfortunately, administration in democracy works with elections, and there was a change in the administration. The whole idea was somehow frozen until the new administration took place, and again took the idea, and again it was welcome. And after a number of ups and downs, the proposal was assessed by international panel in November 2010, and they asked for some revision, although the comments, the overall and specific comments were very positive, and we revised the proposal and finally it was approved. But then we have to translate paper into something else, and this is not very simple. And this initiative, as other which were done in parallel by other scientific environments, was created under the auspices of the university and the Basque government. Things have to be developed, regulations, etc. So it took quite a time to solve those problems, but anyhow, as you see here, the initial operations and the first meeting of the Board of Trustees occurred before the summer, and we are happy to come to today here and show you what we plan to do. Organizations have a mission, and words are a bit perhaps excessive to say what we want to do, but these are the work. So the center has been fostered by the Basque Foundation for Science, Ikerbasque, and the University of the Basque Country, and is going to be devoted to fundamental and translational research with an emphasis on the neuroglia biology. with the aim of discovering ultimately new therapies for brain diseases, and of course, done in cooperation with the local community. I want to emphasize this. This is very important. Our initiative comes from all the neuroscientists in our environment, and naturally networked with international renowned institutions all over the world. And an important aspect of our center will be the training of new generations of neuroscientists. The vision of the center in the midterm will be to strive to lead and coordinate the neuroscience community in the Basque Country by establishing a world-class research center with great research groups and state-of-the-art equipment and facilities. This is very important, people and infrastructure, and to contribute to the understanding on the human brain for making the emphasis on how neurons and glia communicate, and ultimately to generate knowledge which improves the quality of our society. So specifically, the research objectives are to study neuroglia interactions in health and disease, to investigate how neuronal circuits are organized and process information, to characterize the genetic, cellular, and molecular mechanisms which are altered and relevant to neurodegeneration, neuroprotection, and tissue repair, and to develop new therapies to treat the CNS, central nervous system diseases undergoing acute and chronic tissue damage, including Alzheimer's disease and Parkinson's, multiple sclerosis we have before, stroke, and traumatic damage and other conditions. So the organization. We are currently about 50 professionals, which are decisions are taken by the Board of Trustees under the advice of this International Scientific Advisory Committee, a panel of experts I will briefly introduce you later on, and implemented. The decisions are implemented by a scientific director, myself, as of today, who supervises the activity of the research groups, the research platforms, and the administration, which is run by the general manager, who is Jaime Sagardui, who has been in charge of organizing this session. So the Board of Trustees is formed by, supervised by the Department of Education of the Basque Country, and informed by 5 people. 2 representatives come from Ikerbasque, and 2 other representatives from the University of the Basque Country, and 1 from the Foundation, Basque Foundation for Innovation and Health Research, emphasizing this aspect, the will of the new center to interact with the clinicians and to find common grounds to progress in our research. This is the Scientific Advisory Board I was mentioning before. The panel of experts is formed by 10 people, and we have been taking care in balancing national experts and international experts, in particular the US experts and the ones based in Europe. We are happy to have here today 2 of them, Jesus Avila and Helmut Kettenmann, who will give us our plenary lecture. Needless to say that all of them are great experts in the topic of the center. The staff is constituted by 8 groups, and we have here a picture which how this started, that we took in July when we started operations. So the group leaders are Alexei Verkhratsky, José Julio Rodríguez Arellano, Koen Vandenbroeck. We have a chance to talk to listen to him and his excellent research project. José Luis Zugaza, Amanda Sierra, Juan Mancinas, Florence Perrin, Alfonso Araque, and myself. So I would like, and the general manager is Jaime Sagardui. I would like to review briefly what are the activities of the groups. So Verkhratsky's group, who is in turn adjunct scientific director, has a parent appointment with Ikerbasque, and he is based in Manchester, and his research focus is neuroglial signaling in physiology and pathophysiology. José Julio Rodríguez Arellano is an Ikerbasque professor who has been here already for more than 2 years, and his group is named Functional Neuroanatomy Group, and the research focus is neurogenesis and glial alterations in Alzheimer's disease. Koen's group, we already have a chance to listen to the type of work he's doing. The name is neurogenomics. He's a Ikerbasque professor. Indeed, as I was mentioning at the beginning of the session, he was the first Ikerbasque professor among all the ones which are now in the Ikerbasque Foundation, which are around 120 people. So the topic of his research is biomarkers for conditions with neuroinflammatory components, including multiple sclerosis and also stroke damage. José Luis Zuaza is an Ikerbasque professor also located at the Department of Genetics in the School of Science in the campus of Ochoa, and his group is named GTPases and Neurosignaling, and he has focused on how these molecules, together with the molecules of the ROP family, signal or are altered in neurodegenerative disorders, including Alzheimer's disease. Amanda Sierra's group is a group which was formed 1 year ago when she joined us, and the laboratory is the Glial Cell Biology Laboratory, and the focus of her work is microglial cells, in particular how they trim the neurogenic niche in conditions which are normal and in pathological states. Juanma Encinas, similar to Amanda, joined us 1 year ago. He's an Ikerbasque professor, and the group name is Neural Stem Cells and Neurogenesis, and the focus of his research is to examine how neural stem cells differentiate and produce new neurons, and how this process is regulated, in particular, in one of the few niches of neurogenesis, which is the hippocampus and in the adult brain. Florence Perrin leads a group, the name is Integrative Biology of the Generation, and she has concentrated mainly in the spinal cord and how neurons and glia crosstalk in models of injury, traumatic injury, an important health problem, and also in amyotrophic lateral sclerosis. And finally, Alfonso Araque, who has been close to the whole initiative and who was selected in one of the calls of the Ikerbasque Foundation, still to be ascribed to the center. This is in process, and as you heard in his presentation, he has focused on astrocyte neurons interaction in physiology, but also has interest in pathology. Finally, our group, Laboratory of Neurobiology, is focused on studying molecular and cellular mechanisms of neurodegeneration and neuroprotection in vitro, in animal models of disease, including Alzheimer's disease, multiple sclerosis, Parkinson's disease, and finally stroke. And I want to bring here all the senior members of the group who are at the same time project leaders and who are the soul of our group. We also have visiting professors. We had a number of them before, and we currently have 2 of them under the generous sponsorship of Ikerbasque. Robert Sorek is from the University of Ljubljana in Slovenia, and the host laboratory is functional neuroanatomy, and his topic is exocytosis in astrocytes. Astrocytes release neurotransmitters. He is particularly interested in knowing the details, the molecular details. And Rogelio Arellano, who comes from the University Nacional Autónoma de México in Querétaro, Institute of Neuroscience, and who is hosted in the Laboratory of Neurobiology. And the research activity he is doing here is to study adenosine receptors in oligodendrocytes in health and disease. I want also to mention what are the facilities we have now. We are mostly based in the Vizcaya Science and Technology Park, in particular in Building 205, where there are 6 laboratories which are furnished, and work is being done there for a number of years already. We also have a conference room. We are developing core research facilities. I will present you what we have now and what we intend to have in the near future. And there is laboratory space and the management offices. And of course, you all are invited to come and visit us as soon as you can and you wish. This is what we have now, but our plans are ambitious, and there is an agreement between the Basque government and the University of the Basque Country that the final location of the Achucarro Centre will be in the Science Park in the Ochoa campus, and in a building which has this name, Animal Biotechnology Center, which is going to be constructed in a few months, and which will be available in 2015. This is an outlook of how it will look. This building is located at the northern part, or will be located at the northern part of the campus, across the School of Teaching and close to Instituto Barandiaran. And when completed, it should have close to 2,000 square meters, and we intend to have 15, 16 research groups fully equipped and productive at this time. The core facilities we have developed so far is the imaging unit with a super resolution microscope, which is the first of its kind in Spain, and which has a spatial resolution below 100 nanometers. So this is roughly 4 times the resolution of a conventional confocal microscope. And it has a number of features which are very important, such as the fast acquisition of images, which is critical to examine live samples, live preparations, and plus has a high versatility because there is a wide laser which behaves like a radio set in which you can tune the wavelength you want to excite your fluorochrome, and then it opens a wide range of opportunities for the new fluorochromes to be developed. The managers of this facility are Asier Ruiz and Alberto Perez. We plan a number of core facilities for the strategic period of 2013-2016. And these, among other things, the most prominent equipment are the two photon microscope to monitor events in live samples, slices, and also in animals in vivo. And this technology permits deeper tissue penetration, and at the same time is less photodamaging the samples which are studied. We also plan to have a so-called FACS, or fluorescent activated cell sorter, which is important to select and analyze cell populations in many kinds of studies, including studying neuroinflammation and neurogenesis. We also plan to have some imaging system, preclinical imaging system, optical, to monitor whole brain in experimental animals, and also to have a setup for cellular bioenergetics assays in which we can monitor mitochondrial function in health and also in neurodegenerative conditions. So this is the whole thing we have is to provide you with these coordinates in which you can visit the website and see what it is in there. And we also have all these social networks in which you can communicate with us in any way. And just to leave you with this knowledge to cure in Latin, Scientia y Remedium, Ciencia y Remedio. And thank you for your attention. While the presentation is being settled, I would like to briefly introduce Professor Kettenmann. He is a doctor in biology at the University of Heidelberg, and very, very soon, in a few years, became full professor. And he has done many important things in relation to glia research. Among them, perhaps an outstanding contribution was to find that these cells have responses to neurotransmitters. In other words, that they have mechanisms of communication which were thought previously be unique of the neurons. He has been very, very active in the glia field, and he has been the chairman of the Federation, European Federation of Neuroscience Society for a number of years. He has been leading the whole neuroscience community in Europe, and more recently has devoted his work to the biology of gliomas. Gliomas are the unique tumors of the brain, and they have a terrible prognosis. So he's very much interested in studying how to control the growth of these cells and to develop new therapies. And I wonder whether this is coming.
I wonder too.
We apologize for these dysfunctions, which were settled before the start, but then.
copy it on a pen drive?
Takes me some time because there are movies and everything in it. Or I can do it without movies.
Solo detecta que hay una de estas y lo pone. O sea, así como en esto sí que. Igual creo que se ha fastidiado, pero el mío tampoco funciona. Pero bueno, si funciona con este. Si funcionara con este, no. O sea, si estuviera el cable mal, no funcionaría con este. No sé. Será algo de configuración.
Seguro que lo usaban en otros proyectos.
Ya, no, sí, pero estas cosas ya sabes.
Sí, no es la
O sea, es que no me funcionó. Ayer lo tuve. Ayer y a mí. Y de repente me falla.
It. Maybe you switch it off without, because I can see the. I can see the presentation. I guess. Okay, here it is. So what is this? Is this video off?
Here's also problems with it.
Could you lend us your USB drive so we can check? Could you copy your presentation to Alex USB in case somehow my.
But I copied it on.
Yeah, but since it appears damaged, just in case this is broken. I don't know because.
Oh, you mean your USB could be damaged?
in general, and I also want to show some examples of our research. Now, we already heard much about the astrocytes, so in this talk I will focus on two other types of glial cells, namely the microglia and the oligodendrocytes. And in the next slide, just some general ideas about oligodendrocytes. Oligodendrocytes are specific in the white matter, but you also find them in the gray matter. And it lasted until about 50 years ago, until it was recognized that oligodendrocytes in the central nervous system are the myelin-forming cells. For many years, people had thought myelin, which is surrounding axons, was secreted by the axons, and it was the advent of the electron microscopy that researchers learned that there are connections between the oligodendrocyte cell body and the myelin sheath, and you needed to do reconstructions in the EM, and this was done in the 1960s. If you look here, for instance, at that picture, this is the myelin, this is the oligodendrocyte, you don't see the connection, so you have to make a 3D reconstruction to see that it's the oligodendrocyte which form myelin. Now, myelin, as many of you know, is enwrapping axons very tightly, and it is responsible for the saltatory nerve conduction, for the high conduction speed of the nerve, and which reaches a speed of up to 200 meters per second. Now, I think this was the greatest invention of the vertebrates, and this is the prerequisite. Then we can build these highly complex brains as we have in humans. And this was invented by the vertebrates somewhere around 500 million years ago with the advent of the vertebrates in evolution. Why is this such an incredible step? Now, there are enwrapments in invertebrates also, which leads to higher conduction velocity. But this invention of these proteins, these specific proteins which form myelin, like myelin basic protein, myelin associated protein, P0, this was invented by the vertebrates. And this is why they could build connections which are way faster than in invertebrates. I just want to give you an example. In the optic nerve, all your axons are myelinated. You have about 1 million axons in the optic nerve. Now, there is one way you can speed up the action potential by increasing the diameter of the axon. And this is done in invertebrates, for instance, in squid. You know, the squid giant axon has a diameter of about 1 millimeter and reaches the same speed than a myelinated axon in vertebrates. However, if you would like to build an optic nerve with 1 millimeter diameter axons, you would end up exactly like this. This would be the optic nerve. It would have a diameter of 75 centimeters, and it's clear that there is no way that would fit into your skull. Now, the optic nerve is only a small part of the myelinated part. In humans, more than 50% of the brain are myelinated pathways, and you can imagine that it's essentially impossible to have all these connections without myelin. So this is, I think, why I wanted to show you the importance of that evolutionary step, inventing myelin for building these highly these highly complex brains. Now, we had been interested in oligodendrocytes, and I just want to show you a few examples of our recent research. We are actually interested in coupling between the different glial cells in the white matter. And coupling means the expression of specialized proteins called connexins, which form connections between cells. So these are not ion channels which conduct to the extracellular environment, but these are connections between cells. And connexins are primarily expressed in glial cells, particularly in the astrocytes and in the oligodendrocytes. There are specific connexins for the different types of glial cells. And in the oligodendrocytes, it's primarily connexin 29, but most prominently connexin 32 and connexin 47, which is expressed. In the astrocytes, there are other connexins. This is connexin 30 and connexin 34. And we had been studying coupling between the different glial cells and trying to identify the types of connexins which are responsible to connect these different type of networks in white matter. And for instance, we found that coupling between oligodendrocytes depends on the expression of connexin 47 and connexin 32, mainly of connexin 47, and the interconnection of oligodendrocyte to astrocyte coupling depends on 47 on the oligodendrocyte and connexin 30 on the astrocyte. Now, is that really important for brain function? That is an essential question. And indeed, actually it is. Because if you have mutations in these connexins, you find that there are problems with myelin formation. For instance, we had been studying mice which are deficient of connexin 47 and connexin 30, and this eliminates the communication, the connexin communication, the gap junction communication between astrocytes and oligodendrocytes. And what you see here is that half of the mice die late in development. After about 30 days, they are starting to die. Some survive. So obviously, these connexins, these interconnections between the glial cells are very important for that important function, namely myelin formation. And that these connexin 47 and 30 deficient mice are hypomyelinated is shown here. So we analyzed the thickness of myelin in the heterozygous and in the knockout mice. And when you do the statistics, you find that these mice really have a problem with myelin and myelination, and this is why they are impaired. We also studied actually a point mutation in connexin 47, which was found in a family which has cases of leukodystrophies, very severe impairments. And these patients, which are affected by these point mutations in connexin 47, develop severe impairments, and they also have a very strong hypomyelination. Actually, we made a mouse model, which we published last year, where we induced that point mutation into the mouse, and that mouse has the same problems. So it's essentially a problem. So myelin formation really very much depends on the communication between these different types of glial cells. Now, let me switch gears and come to the second part of glial cell, which I would like to introduce to you, and these are microglial cells. Now, microglial cells are also abundant everywhere in the brain. There is no region in the central nervous system which is devoid of microglia, including the retina, the optic nerve, and the spinal cord. So microglial cells are essentially everywhere. What we know about microglial cells is that they are, of course, present in the normal brain. They are in a ramified form, and they were termed in the old days, now we are changing this concept, the resting microglia. Now we know they are not at all resting. They are highly active in the normal brain. But what was established for a long time by pathologists is that if there is any injury to the central nervous system, these cells respond. And they respond with a quite interesting program, which we call activation. I'll come to that concept a little bit later. So they can actually actively proliferate, actively migrate to the site of the injury. In the pathologic brain, they change their form, become amoeboid. They can release a number of cytokines and chemokines, growth factors. They are the antigen-presenting cells of the central nervous system. And they can actively phagocytose, and I will come to that. Now, actually, when Alex showed this first image, when glial cells were described by Rudolf Virchow. And when you actually look at these cells, which he described for the first time, terming glia, if you look at these cells look exactly like activated microglial cells. So what he probably first said were not astrocytes or oligodendrocytes, but probably activated microglia. And actually he was a pathologist, so he was really looking at the pathologic brain. And so I think microglial cells were the first glial cells which were discovered. It was already recognized in the early days of the pathologists that associated with diseases, you find these activated cells, and these are drawings made by Alois Alzheimer. I will come back to Alois Alzheimer a little bit later, and he depicted here these activated cells, and these are most likely activated microglial cells. However, in those days, people did not distinguish between the different types of glial cells. One talked about glia. The term astrocyte was invented, but it was not really clear what an astrocyte was. Actually, we even today do not know precisely what an astrocyte is. But the distinction between the three different types of glial cells in the central nervous system had to await until a student of Ramon y Cajal defined it. And this was, as you can see, Rio Ortega, and he defined the three types of glial cells in 1919. And he actually laid the concept of what we knew for many years about microglial cells. Actually, his studies, starting in 1919, were summarized in a fantastic review he wrote in the Penfield article in 1932, and much of our knowledge, until about fairly recently, was based of what Ortega described in this review, and I just want to go very briefly through what we know about microglia. Now, it was evident that these cells are intruders. They invade the brain early in development, and since very recently, we know that these cells actually stem from the yolk sac and invade the brain very, very early in embryonic times. They come in also later, and it's at the moment very heavily debated how much exchange there is, and it may be even true that you still live with your microglial cells which had invaded into your brain in your embryonic life. Now, they are pretty motile and move around, and actually I hope I was really quickly copying over some of the movies. I'm not so sure how many I got over and whether it works, but you will see they are highly motile. They are the highest motile cells in the central nervous system, really moving around pretty quickly. Now, in the normal brain, they differentiate into the ramified microglial cell, and you see that characteristic morphology here, and you see on this overview in the hippocampus that they're all over. There is no region in the brain where they don't have actually a kind of a territory. Now, it's also evident that this activated glial cell, which you see in a pathologic region, is a transformation of the resting ramified form into this activated form, and this was shown by Ortega here. Here you see the ramified form, then the processes are becoming shorter, and then they are acquiring this amoeboid morphology. And they really accumulate at the site of injury. And what he already showed is that they really associate with damaged neurons and even having indications that they might phagocytose them. Now, actually, I'm surprised it works. This is how rapidly microglial cells actually move in a dish, in a culture dish. And you see here actually the hours, minutes, and seconds going by. And when you look at that, you really see that within seconds they can move. Actually, when you sit in front of the microscope and focus on their processes, you can really in life see them moving. So it's a very rapid movement. Now, this is another example also from the cell culture where we have a substance which attracts microglia. And you'll see here that some of these microglial cells are just moving towards that pipette where a chemoattractive substance is released. It's actually complement factor 5A, and you see this cell is just pretty rapidly moving to this source. So these cells can really rapidly move through the brain and respond to a large number of chemoattractive factor. One of these examples is this complement factor 5A. This probably does, oh man, I don't know what this means. No? Okay, good. Actually, that's a slide is an image in Vivo where we are recording, and actually Van Kirchhoff was the first who demonstrated that these microglial cells are extremely rapidly moving, even in the normal brain. They constantly scan the brain, but I'm fairly sure that that movie won't work because I didn't copy this audio, but I didn't have time enough for that. Now, coming to phagocytosis, and I just want to show you, I think that works again. I show you that these cells are really the professional phagocytes of the brain. And when I run this movie again, I would like you to focus on that microglial cell here. And this is actually from a sliced surface where they are activated microglial cells. And you will see that this cell here, this microglial cell, moves around with their arms, and then it finds something which it likes and pulls it close and even incorporates it and eats it. And this whole process lasts only a few minutes. Another example is when you have a stroke and blood is entering your brain, erythrocytes for instance, and you see this microglial cell is just eating an erythrocyte, and I just run this again that you can see it again. And you see this cell here eating an erythrocyte here. And it slurps it away. And as you see this process, these are the minutes passing by, and the seconds, it's over in a few minutes. If you imagine that you all of a sudden activate your microglial cells to their full phagocytic capacity, I think you would not have time to leave the room before your brain is probably eaten. Microglial cells in defined diseases. And wait a minute, I'm missing a slide. Damn it. I'm missing a slide, but I describe it to you. And this was actually a summary slide which we had published in a review a couple of years ago with Uwe Hanisch. And this formulated the concept that microglial activation is not an all or none process, that it's not something which is like an action potential, switching on or switching off. But microglial activation is a very diverse process, and depending on the pathologic context, it can be very distinct and very diverse. Now, when you look at the morphology, they look pretty amoeboid under all kinds of pathologic conditions. But I think one should not confuse the morphological change and this morphological transformation into the amoeboid form into functional phenotypes. And we are getting more and more evidence that microglial cells behave quite differently under different pathologic conditions. And I just want to show you at the end of that lecture two examples how microglial cells can be quite different as one would expect it actually. And also during the pathologic process, these cells change. Now, most people think that microglial activation is microglial cells with a pro-inflammatory phenotype. They release all these pro-inflammatory cytokines, chemokines, such as TNF-alpha, interleukin-6, for instance, nitric oxide. So they are these bad guys who kill cells, eat cells, proliferate, and just conquer the brain. Now, we had been actually interested to look at microglial cells in the context of Alzheimer's disease, and this is actually the first new pathological description by Alois Alzheimer of the disease. And what you see here are actually three important hallmarks he already recognized in his publication in 1911. When he described the histopathology for the very first time. There are the plaques, which are very important. There are the damaged neurons, which he called ganglion cells at that time, so he recognized neurons are damaged, and what he recognized were the GA sets, and these are the activated glial cells. So he recognized that around these plaques there are activated glial cells. Now, we had been studying these cells in the live brain, and we had used an animal model of Alzheimer's disease, and actually asked the question, when these cells are associated with the plaques, how is their function being altered? And what we had been looking was the movement of the processes and their phagocytic capacity. What you actually would expect is that when they are in the Alzheimer environment and they are amoeboid, that their phagocytic activity would increase, because this is an activated microglial cell, and an activated microglial cell makes phagocytosis. What we essentially found was just the opposite. And I don't want to, I just give you the main message, and that is, once the plaques appear, these cells become dysfunctional, and their capacity to do phagocytosis decreases by about 50%. This is shown, for instance, in the cortex of these mice, and in the cortex of these mice, the plaques develop at about 8 weeks, and once these plaques appear, then the phagocytic activity goes down. In these mice, actually, in the hippocampus, that occurs later, so the plaques appear later in the hippocampus, so there is no change in phagocytic activity in the hippocampus at about 8 to 9 weeks, because there are no plaques. But plaques appear at about 4 months in the hippocampus, and then the phagocytic activity goes down. In the cerebellum, for instance, plaques do not appear, and this phagocytic activity stays as in a control. So this is an example where, quite unexpectedly, these cells are called activated microglial cells, but one of their functions, and that is phagocytic activity, is even downregulated, and in contrast of what one would connect with an activated microglial cell. Now, in the last few slides, I would like to show you another brain disease which we had been interested, and this is gliomas. Now, most people do not realize that if you take glioma tissue out of a human patient, this tissue is filled with microglial cells, or brain macrophages. Up to 30% of this glioma tissue consists of brain macrophages, and this is a mixture of intrinsically invading microglia into this glioma tissue and also infiltrating macrophages from the periphery. Now, what is quite evident is that these cells acquire the amoeboid morphology, so you would think they are activated microglial cells. However, what's also evident, and this we know, of course, from the patients, that these cells are not at all eating up the gliomas, otherwise they would have been gone. Now, in a series of studies, and I just want to show you 2 or 3 more slides, because I'm your main obstacle between your lunch, so I have to speed up, is that actually microglial cells are pro-tumorigenic. Microglial cells help the glioma cells to invade into the brain and expand and grow. And we have shown that in vitro preparation, but we also have shown that in an in vivo preparation in collaboration with Frank Heppner. So he had developed a mouse model which you can deplete of microglial cells. This is a mouse which expresses a thymidine kinase under the control of a promoter which in the brain is specific for microglia. This is the CD11B promoter. This means this thymidine kinase in the brain is only expressed by microglial cells. Now, if you then apply ganciclovir, then this kinase is activated and the cells die. So within a few days, microglial cells are strongly depleted in this mouse. So what we did was we implanted glioma cells, then applied after 7 days ganciclovir, depleted microglia, and then we looked at these brains. And what you see here, this is the tumor volume in the control. These are the treated mice. Here the microglia cells are depleted, and these tumors are much, much smaller. And of course, there are much less microglial cells. What is the mechanism by which microglial cells help glioma cells to invade into the brain? And I just have only a few, wait a minute, actually a summary slide, and this is based on several publications which are partially out already and which are just coming out. Now, what we learned is that glioma cells reprogram microglial cells into a phenotype which helps them to invade. Now, the glioma cells release a factor or factors which triggers a response in the microglial cells via a receptor which we have recently identified, and this is just submitted, and these are toll-like receptors, TLRs, receptors which are normally recognize danger signals. But we also know in the meantime there are a lot of endogenous signals which can be released and activate TLRs, these toll-like receptors on microglial cells. Once this receptor is activated, and it needs an adapter protein here, then it activates an intracellular cascade and leads to an expression of an enzyme which is not expressed under normal conditions in microglia, so it's only induced when they are confronted with gliomas. And this enzyme is called MT1-MMP, or also MMP14. It's a metalloprotease which cleaves other metalloproteases. Now, the tumor actively releases also metalloproteases to degrade extracellular matrix, that it can infiltrate into the brain. But it releases these metalloproteases in a longer form, which needs to be cleaved to be activated, and one prominent one is pro-MMP2, metalloprotease 2. Now, this enzyme actually cleaves it and really activates it, and so the glioma cells reprogram the microglia that they express an enzyme which helps them to degrade extracellular matrix and invade into the brain. And we have done a whole series of experiments. For instance, if we knock out MT1-MMP, then the tumors are much smaller. If we interfere with the intracellular pathway, the tumors are smaller. And here I'll show you an example where we knock out TLR2 on the microglial cells, and this is in the experimental animal model, tumor size in the control and tumor size in the knockout, and you see the tumors are much smaller. Lastly, this can be also clinically exploited, and I just show you in this last data slide that an antibionic minocycline interferes with that intracellular pathway, this P38 MAP kinase, which is necessary to signal from TLR2 to the expression of MT1-MMP. And when you actually apply minocycline into the drinking water of mice and inoculate tumors, and this is the animal model, just look at this example here, that is the control. This is the tumor size of the minocycline-treated animal, and then you see the tumor sizes are much smaller, and we are at the moment have been written a proposal, a grant proposal for doing a clinical trial, and hopefully this may be of benefit on the long run also for the patients. Now, at the end, of course, this work was done with a lot of collaborators, which are listed here, and this is actually the center in Berlin-Buch where I had been working. Thank you very much. Lunch, huh?
Yes, to thank you for your attention, for being with us today. And we would like to invite you now for a cocktail, which has been supported by Novartis, who is sponsoring this event. So thank you again, and we are in touch.
for you.
Thank you.